Melangkah berjalan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan MKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan MKH semakin hebat. Melangkah berjalan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan MKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan MKH semakin hebat. Melangkah berjalan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan FKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan, sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan FKH semakin hebat. Melangkah ke depan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan, sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan FKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan, sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan FKH semakin hebat. Melangkah ke depan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan, sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan FKH selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan FKH semakin hebat. Melangkah perjuangan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan FKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan FKH semakin hebat. Melangkah perjuangan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan FKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo,
Namanya udah gede tapi
Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan MKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan, sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan MKH semakin hebat. Melangkah berjalan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan, sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan MKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, selalu di depan, junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan MKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan MKH semakin hebat. Melangkah ke depan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan, junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan MKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan MKH semakin hebat. Melangkah ke depan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan, junjung etika moral, jaga lingkungan lestari. Ayo, selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan FH semakin hebat. Melangkah ke depan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternak dan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan FH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternak dan sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan FH semakin hebat. Melangkah ke depan di Indonesia dan di dunia. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternak dan sejahterakan masyarakat. Universitas Airlangga,
Hello, can you hear me?
di depan.
Yes, Dr. D.
Okay, very good. Just testing the mic. All right, thank you.
You're welcome, Dr. D.
Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, selalu di depan, junjung etika moral, jaga lingkungan lestari. Ayo, ayo jadikan FKH semakin hebat. Sarjanamu, dokter hewanmu, siap membangun negeri. Tekuni, teliti, ilmu veteriner dan teknologi, inovasi kesehatan hewan peternakan, sejahterakan masyarakat, sejahterakan masyarakat. Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo jadikan FKH semakin hebat. Melangkah ke depan di Indonesia dan di
Halo, selamat malam, yo.
Selamat malam, Prof.
Ya, oke.
inovasi kesehatan hewan peternakan, sejahterakan masyarakat.
Halo, selamat malam, yo.
Selamat malam, Prof.
Ya, oke.
Inovasi kesehatan.
Halo, Dokter Lee. Halo.
Hi, can you hear me?
Ya, halo, Dokter Lee. Ya, my name is
Hai,
Mubasirin.
selamat malam.
Selamat malam. Ya, my name is Mubasirin. Ya, from Parasitology Division. Ya, Faculty of Veterinary Medicine. How's the weather in Malaysia?
Pardon? I can't hear you clearly.
Oh, we already experiencing rainy, yeah, weather in Surabaya.
Same here. The weather has not been very good of late. Yes.
Okay.
So that may affect the internet connectivity by a bit.
Okay. And there have been flood in some area in Surabaya, yeah. Oke. Halo, Prof. Endang. Good evening. Mood, Prof. Endang.
Halo, Assalamualaikum.
Waalaikumsalam.
How are you, Mr. Lee?
I'm fine, thank you. How are you?
Iya.
I'm fine, thank you. Alhamdulillah, visit in a quiz lecture.
Happy to join you guys.
Dr. Lee, my division have six lectures ya, including Prof. Puji. Halo Prof. Puji, Prof. Endang,
Ya,
Prof. Endang ya, Prof Lucia, ya, Prof Yunus, Prof Yunus Makkah ini ya. Miss Yao from Malaysia, student, Dr. Lee. Halo Yao,
Oke, Prof.
ya.
Selamat malam.
Oh, good evening, Prof Lucia.
Good evening.
Hi, Prof. Good evening.
Good evening, Dr. Wen Yuli. Nggak usah.
Hi. Nice meeting you all.
Nice to meet you.
Nah, good you.
Thank you for your willing give us guest lecture.
It's a pleasure and honor. Thank you so much for inviting me over.
You're welcome. I heard that you can also speak Melayu. It's true?
Sedikit.
Oh, sedikit.
Little, sedikit-sedikit.
Little bit. Sedikit.
Well, after all, Bahasa Malaysia is very similar to Bahasa Indonesia. So I
Yes,
think
yes.
we share a lot of vocabularies and cultural heritage. So I think conversation wise, the barrier is definitely thinner. And yeah, so I think citizens from both countries can understand each other pretty easily.
Yes.
Yeah.
Sudah jam 7, Pak Mufa.
Miss Yao, please,
Baik.
start ya.
Rif,
Ini
disiapkan,
sudah
Rif.
jam 7, so saya akan mulai pembukaan event acara ini. So, selamat malam, salam kebajikan semua. Good evening, everyone. Greetings to our honorable heads of Parasitology Department, Dr. Mufasirin, Dr. Hewan, Master of Science, our respected moderator, Prof. Dr. Lucia Tri Suwanti, DVM, Master of Agriculture, dan semua dosen parasitologi yang kami hormati. Our distinguished guest speaker, Dr. Wenn Chyau, Ph.D., and all our beloved Faculty of Veterinary Medicine students. Welcome to the guest lecture of Parasitology Veterinary with the topic The Sticky Business of Plasmodium, a dive into malaria immuno-pathobiology. So each participant are encouraged to turn on your cameras as a respect to our guest lecture. Before we start our acara, let me introduce myself. My name is Lim Chyau, as a parasitology practical assistant this year, and I will be the master of ceremony for today's event. Now let's begin today's session by singing together the national anthem of Indonesia, Indonesia Raya.
Silakan Arif.
Nggak ada suaranya ya.
Indonesia tanah airku, tanah tumpah darahku. Di sanalah ku berdiri, jadi pandu hidupku. Indonesia kebangsaanku, bangsa dan tanah airku. Marilah kita bersatu, Indonesia bersatu. Hiduplah tanahku, hiduplah negeriku, bangsaku, rakyatku semuanya. Bangunlah jiwanya, bangunlah badannya, untuk Indonesia Raya. Indonesia Raya, merdeka, merdeka, tanahku, negeriku yang kucinta. Indonesia Raya, merdeka, merdeka, hiduplah Indonesia Raya. Indonesia Raya, merdeka, merdeka, tanahku, negeriku yang kucinta. Indonesia Raya, merdeka, merdeka, hiduplah Indonesia Raya.
Baik, so for now it's our first documentation session. So everyone is encouraged to turn on your camera with the virtual background that given. Okay, so I will start from the first slide. 1, 2, 3. The second slide. 1, 2, 3. Third slide. 1, 2, 3. The fourth slide. 1, 2, 3. The fifth slide. 1, 2, 3. The sixth slide. 1, 2, 3. The seventh slide. 1, 2, 3. The eighth slide. 1, 2, 3. The ninth slide. 1, 2, 3. The 10th slide. 1, 2, 3. And the last slide. 1, 2, 3. Okay, so now we are having to move on to the introduction to our guest speaker and reading of his curriculum vitae. And this session will be led by our moderator, Prof. Dr. Lucia Suwanti, DVM, Master of Agriculture. Prof. Dr. Lucia Suwanti, DVM, Master in Agriculture, is a professor in Division of Veterinary Parasitology in Faculty of Veterinary Medicine, Universitas Airlangga. She has completed her veterinary medicine degree at Gajah Mada University and earned her master in veterinary science from the same university and also obtained her doctorate in medical science from Universitas Airlangga. Prof. Lucia is an active researcher in zoonotic and non-zoonotic parasite research group and the toxoplasmosis study group, focusing on parasitology and immunology. Prof. Lucia has published more than 50 international papers indexed by Scopus, highlighting her strong contribution to the field of veterinary parasitology. To Prof. Lucia, the time is yours.
Thank you, Lim Chyau, for introducing me. Good evening, ladies and gentlemen. professor, lecturer, student, and of course our guest lecturer, Dr. Wenn Chyau, Ph.D. I will like to express my gratitude for being interested to moderate this guest lecture with title The Sticky Business of Plasmodium, a dive into malaria immuno-pathobiology. Before Dr. Wenn Chyau present, I will read his curriculum vitae. Dr. Wenn Chyau, Ph.D. was born in 1988. He is still young, the same age as my daughter, so I think it's still young.
Yeah.
Thank you, Doctor. It's very I think it's interesting changes to heard your presentation. Before it, I will read your curriculum vitae. Dr. Wichu, PhD, received his BS degree in biomedical sciences from University Malaya, Malaysia in 2011, and graduated with his PhD in medical sciences also from University Malaya in 2014. He started his postdoctoral research career at the Agency for Science, Technology and Research, ISTAT, of Singapore. He joined the Department of Parasitology, Faculty of Medicine, University of Malaya as a senior lecturer in December 2021. Currently, he also an adjunct investigator of START ID Labs, Singapore. His main research interests revolve around the immuno-pathobiology of malaria, with a special focus on cytoadherence phenomenon by the parasite red blood cells. He also involved in studies of biological factor control, focusing of Wolbachia-based strategy. And to date, he was published as a first or also corresponding author in reputable journals such as Blood, Frontiers in Immunology, Frontiers in Cellular and Infection Microbiology, iLife, Biomedicine, Entomologia Generalis, and Trend in Parasitology. I think it's more than it. Currently, he is also associate editor of Frontier in Microbiology. Dr. Lee, now screen and time is yours.
Thank you, Prof, for the kind introduction. It's a pleasure and honor to be invited by Universitas Airlangga for this guest lecture. So I would like to share my slide. Hold on, yeah. Can you guys see my slide?
Loading still. Oh yes, okay.
Okay. So, all right, let's get started. So today I would like to talk to you on this cytoadherence phenomena by Plasmodium infected erythrocytes, which is closely associated with the immuno-pathobiology of malaria. Um, just to get started with something light. So malaria is a mosquito-borne diseases caused by blood parasites of the genus Plasmodium. And I'm pretty sure Indonesians are quite familiar with this disease. So I don't need to do much introductions about these parasites. So there are currently quite a number of medically important malaria parasites as listed by this flow chart here. So I actually categorize them into human malaria parasites and zoonotic malaria parasites. So those highlighted in bold are definitely the parasites with increased significance in terms of healthcare burden. Now, of these, Plasmodium falciparum is the most prevalent malaria parasites on the African continent, and is also the most fatal species among the medically important malaria parasites. On the other hand, Plasmodium vivax, which is the one shown on the second column on the left, is actually the predominant species in most countries outside of Africa. And among the zoonotic malaria parasites, especially for Southeast Asian region, Plasmodium knowlesi, the first parasites listed on the right, is actually gaining traction due to its persistent transmissions in this region. So a little bit more about the malaria biology. So it remains a massive healthcare burden to many countries actually. And because of COVID, the burden of malaria was actually increased during the COVID period. And although we have seen to successfully bring it back towards the lowering trend, the burden incurred by malaria remains high. And at the moment, we are still waiting for WHO report on the transmission biology of malaria in year 2024. And of course, we hope that the trend is actually following the downward trend. And a lot of people will always associate malaria with Africa, but besides Africa, Southeast Asia is actually the second, the region with the second heaviest malaria burden. And this is actually one of my common research field sites in Thailand. The picture on top is actually Professor Francois Norton, a friend of mine, who is actually leading the Sokro Malaria Research Unit in That Province, Thailand, who overseeing clinics for refugees across the long stretch of Thai-Burmese border. And the picture at the bottom are actually showing the some of the refugee camps along the border. And these areas are heavily hit by various tropical diseases, including malaria. So how is it transmitted? Now, there are four major routes of transmissions for malaria. And of course, the natural transmission mode is actually through mosquito bite. Nevertheless, other modes of transmission, such as transfusions, vertical transmissions, and usage of contaminated syringes, whether in the healthcare setting or due to drug abuse, are actually significant routes of transmissions of malaria as well. Now, whenever we talk about parasites, we always emphasize a lot on the life cycle of the parasites. So this is a simplified schematic diagram showing the complex life cycle of Plasmodium, involving two different organisms as the host, which are the mosquitoes as the definitive host, and us humans or primates as the intermediate host. And then within the human, we can further divide the stages into the liver stage, or we call it the exoerythrocytic cycle of the parasites, and the erythrocytic stage of the parasites. Now, the clinical signs and symptoms that we see in malaria patients are actually attributed to these part of the life cycle, which are the blood stages of the parasites. So how do these particular parts of the parasite stages cause the signs and symptoms seen in malaria patients? So as the parasites matured and developed within the infected erythrocytes, they cause substantial changes to the architectures of the infected erythrocytes by inserting their own proteins onto the membranes of the infected erythrocytes. So one of the examples of proteins that they insert into the membranes of the erythrocytes is actually the PFEMP1, as shown by this schematic diagram here. Now, because of these insertions of foreign proteins onto the membranes of the infected erythrocytes, it caused disruptions to the membrane stability and dynamics of the infected erythrocytes. And these actually alter the deformability or rheological properties of the infected erythrocytes. And in the case of Plasmodium falciparum, it actually makes the erythrocytes more rigid. Now, these create an issues for the parasite survival. So, within our body, we have this organ called spleen. Okay, so this is actually the filter unit for our circulations. So, every milliseconds, the entities. So actually, I put this schematic diagram on the right to show the mechanical workflow of how circulations actually go through the spleen, like a filter unit for the circulations. So all these entities within the circulations, they will have to go through the spleen, and this spleen has this anatomical part known as the rat parts. So within the rat parts, they have these small spaces with gaps that are actually smaller than the smallest capillary of our body, which for adults is actually not more than 2 micrometer in size. So with these gaps of space, uh, is actually smaller than the size of a rat cells. However, normal healthy cells are very flexible. So they are capable of squeezing themselves through this filter unit and get back to the circulations, as shown by this cartoon here. However, defective cells or infected cells or oxidatively damaged cells will become more rigid, and they will get trapped within the spleen rat parts and eventually get cleared by the resident macrophages within the spleen. So if the parasites were to survive and establish within the host, they have to come up with a way to actually overcome these splenic filtrations. So the malaria parasites are very smart. So they in fact come up with a counter strategies to escape our splenic filtration. So what they do is some of the proteins that they actually express onto the surface of the membranes of the infected erythrocytes are very sticky, and they confer these altered cytoadherence properties to the infected erythrocytes. And what happened is that these allow the infected erythrocytes to cytoadhere directly to the endothelial cells. So the picture on the lower right is actually a seeded endothelial cells, and you can see that. Let me find my laser pointer. Hold on. Okay, so you will see that the red cells, the infected red cells, are actually attached onto the endothelial cells. So all these purple color thing here are actually the malaria parasites, and you can see the malaria pigments within the parasites. So they are highly sticky. Sorry about my slides. Okay, so by gluing themselves stably onto the endothelial cells, this allowed the parasites to actually sequester within the vasculature of our circulations. And of course, by doing so, they will be able to escape or prevent themselves from reaching the spleen. And this will escape the splenic filtrations. Now, the side effect of these immuno-escape mechanisms is that the direct adherence of the infected erythrocytes to the endothelial cells of the vasculature will actually stimulate or activate the endothelial cells, which leads to the inflammations of the vasculature. And eventually, our vasculature will become leaky, which is the hallmark of severe pathology of malaria. So that is how the blood stages of the parasites cause the pathogenesis in severe malaria. So what are the package of severe malaria? So when we talk about severe malaria, it is not pinpointed to only one or two types of signs or symptoms. It is actually a collection of complications that can be caused by malaria parasites. And depending on the geographical origins of the parasites, the severe complications caused by the parasites may differ. So these include, of course, the most notorious one, the cerebral malaria, which is actually contributing to a lot of the fatalities associated with malaria. We also have acute respiratory distress syndrome, acute renal failure, severe thrombocytopenia, as well as severe acidosis and severe anemia. And certain locations, certain geographical origins may also have a relatively rare severe complications such as placental malaria. So all these are somehow associated with the cytoadherence phenotypes of the infected erythrocytes. So back to the diagram that was shown just now in the previous slide, you might have noticed that there is this crumb of thing here, which is known as rosette in the picture. Yet it is not really a direct attachment of infected erythrocytes to the endothelial cells. So this is actually known as the rosetting phenomenon. But what exactly is rosetting? So rosetting is actually a phenomenon where the late-stage infected erythrocytes is stably attached to a row of uninfected erythrocytes, forming this flower-like structure, hence the name rosette. And this is the actual pictures of the parasite showing how rosettes looks like under the wet mount. And we actually put dye, Gemsa dye, into the wet mount suspensions to give better color and better visualizations of the phenomenon using basic equipment like a simple light microscopes. So the picture on the left shows simple sized rosettes, whereas the picture on the right shows a giant rosette. And these are basically stable structures that can actually withstand shear force as high as those within artery. So these structures are actually showing better binding stability than the direct attachments of infected erythrocytes to the endothelial cells of our vasculature. But what exactly does rosetting phenomenon contribute in terms of malaria pathogenesis? So since its discovery back in the 1980s in monkey malaria, various roles have been postulated for rosetting in malaria pathogenesis as listed in this particular slide here. However, all these postulated roles were not adequately tested prior to our involvement in this rosetting research, and it all remains as postulations. And in fact, there were certain school of researchers that were very skeptical about role of rosetting in malaria pathogenesis because some of them actually believe that rosettes may not actually be available within the body. So it might just be an artifact induced in the in vitro setting. Nevertheless, when we look at the situations and the story behind rosetting phenomenon, back in 2011, we decided to give it a try to actually investigate further to deduce and validate the roles of rosetting in malaria pathogenesis. So the first thing that we actually tested was the postulated roles of rosetting to facilitate the reinvasions of malaria parasites. So this hypothesis was pretty popular, and it was one of the first few hypotheses that was generated during the discovery of rosettes. And logically speaking, it does it seems to make sense because if you have a row of uninfected erythrocytes surrounding the infected erythrocytes at the center, then when the parasites is matured with the schizones rupturing, releasing the merozoites, then the merozoites will have fresh uninfected red cells readily available in the vicinity for the parasites to reinvade. So in other words, it will help the parasites to replicate much more easily. However, as early as 1998, a research team led by Barbara Croft actually proved that this was not the case for Plasmodium falciparum, where rosettin actually did not facilitate the reinvasions of Plasmodium falciparum. However, the work was actually stopped at the level of Plasmodium falciparum because during that time, there was a lacking of a technique that was suitable to actually do further validations for other species such as Plasmodium vivax. This is because for Plasmodium vivax, the parasites can only invade a certain group of our red cells, which are actually the younger red blood cells, which is known as reticulocytes. So during that time, there was no technique that can actually visualize rosettes and reticulocytes simultaneously. So when we discover this knowledge gap, we decided to give it a try. Okay, so this is how the reticulocytes or the immature red blood cells looks like. So within the cells, you will see this bluish dot thingy, which is actually the reticulated matters or the remnants of nucleic acids and ribosomes that were soon going to be a kick out from the cells during the maturation process of the reticulocytes towards the matured red blood cells, or we call it normocytes. So we managed to develop a technique that can actually visualize both rosettes and reticulocytes. And so this was the technique that we developed back in 2013, where we managed to visualize the reticulocytes as shown on the in the picture on top, and the parasites within the wet mount by just using very cheap dye material, which is GEMSA, and a very simple technique used in the field of parasitology, the wet mount. So by just combining two very affordable methods together, we found that we could actually do this. So we decided to give it a try to test this hypothesis for Plasmodium vivax. And in fact, we found that Plasmodium vivax actually preferably rosette with the normocytes. So this also ruled out the hypothesized roles of rosetting to facilitate parasites reinvasions for the case of Plasmodium vivax. So the work involved in testing this hypothesis was a pretty easy one, where it took us less than 3 years to actually complete the work of testing. So subsequently, when we move on to the next one, yes, the challenges become tougher. So to test the next hypothesis, which is to facilitate the sequestrations of the infected erythrocytes, independent of direct bindings of infected cells to the endothelial cells, we decided to use a combinations of bioengineering technique known as the microfluidic assay and micropipette assays. So let's take a look at this video here. So this is the video showing the demonstrations of micropipette technique, where we applied, apologies for the technical glitch, okay, where we applied force mimicking suction force to suck out the infected and the non-infected cells into the micropipette to separate the rosette. So you can see that on the left, the uninfected erythrocytes, apologies for the technical glitch. Okay, so you can see that the uninfected erythrocytes could actually be aspirated deep into the channel. On the other hand, the infected erythrocytes are simply too rigid to be aspirated into the micropipette of the same size, using the same aspiration force. So by doing so, we can actually evaluate the rigidity of the cells involved, as well as quantitate the binding force involved in rosette formations. And this is microfluidic assay. So take a look at these videos here. So this is a video showing how fast uninfected erythrocytes can move through these microfluidic channels that mimic microvasculature. And the video in the center shows the infected red cells. So it gets stuck a little bit, but eventually it will still be able to flow through the microvascular mimicking a microfluidic channel. And finally, in this video, you can see that when they form rosettes, the whole rosetting complex become more rigid and they simply get stuck at the channel opening. However, do pay attention that while the rosette is stuck, you can see that there are things, there are red cells that can still move through the channel opening, meaning that the channel is not completely blocked by rosettes. So with that, we prove that rosettes can actually confer increased rigidity to the whole structure, including the infected red cells, which help them to mechanically sequester in the microvascular without the need to directly adhere themselves to the endothelial cells. So with that, it will also prevent complete blockage of the microvascular in the host, which may actually reduce the severe pathogenesis of malaria. As if you do not have a complete blockage of microvascular, you may actually reduce the likelihood of localized tissue hypoxia to the affected area. So with that, we actually proof that rosettes can actually help the parasites to sequester without the need to actually gluing themselves to the endothelial cells. So this also means that the parasites can widen their zone of sequestrations within our vasculature. So this work was actually pretty technical in terms of a level of challenging, where we actually need a little bit of engineering technique to prove it. Then subsequently, we move on to the next hypothesis, where the challenge to test it was actually even harder. So the next hypothesized roles of rosetting that we tested was to actually test whether the rosetting could hamper phagocytosis of the infected red cells by the host phagocytes. So again, this was a pretty logical kind of hypothesis because when the parasites form rosettes, they surround themselves with uninfected red cells. So this may actually shield the infected red cells from being recognized by the human phagocytes. So it's like a camouflage. And besides, the rosetting complex is actually larger. So it may make it much more difficult and challenging for a single phagocytes to engulf the whole rosette complex. So with this in mind, we decided to actually test this hypothesis systematically. So along the way, we actually found that the parasites could sense the incoming threat in the form of phagocytes via the protein secreted by the phagocytes. And in this case, it was this protein known as insulin-like growth factor binding protein 7 or IGFBP7. So they sense this protein that was secreted by phagocytes as a signal that the phagocytes are coming. And then they smartly use this protein as one of the bridging factors for them to form more rosettes. And in return, these rosettes actually makes it more difficult and challenging for the phagocytes to engulf, to clear the infected erythrocytes, as shown by the pictures on the right. So as you can see on the left panel here, when the infected erythrocytes do not form rosettes, it is pretty easy for a single monocytes to actually engulf them. So you can see that the schizones has been completely engulfed. Okay, has been completely engulfed by these phagocytes. Okay.
Baterai kok habis.
Can you see the slide?
When
Okay, very good.
Chyau.
Yes,
Yes?
yes, yes.
All right, very good. Apologies for the
Oi.
technical glitch. Okay,
Chyau.
so
That's okay.
however, when the parasites start forming rosettes, a single phagocytes would not be able to engulf it successfully. In return, the phagocytes have to actually recruit more phagocytes so that they can work together just to demolish one particular rosette forming infected erythrocytes. And can you imagine if this is actually happening within our body? It can actually exhaust the pool of phagocytes within our body. So in return, it will actually enhance the survival of the parasites within our body. So this discovery of rosetting serving as a immune escape mechanisms for the parasites against phagocytosis was actually featured in news coverage during its discovery, and it also earned us a few international patents. And not long after our discovery, a separate team from Brazil actually found similar discovery regarding the roles of rosette to hamper phagocytosis of Plasmodium infected erythrocytes. So with that, we actually change the perspective of rosettes in malaria pathogenesis, where we also recategorize the types of rosettes in malaria, where we divide them into three types based on the interactions and the requirements of the protein involved, whether it is just a simple interactions between receptors and ligand, or where the host derived proteins are needed as a bridging factor to actually form the rosettes, which is what we know as the type 2 rosettes. And the IGF-BP7 that we discovered was actually considered as the contributor towards the type 2 rosettes. And of course, we have the type of rosettes where um, the rosetting ligands involved are actually not securely expressed onto the surface of the infected red cells, where they can actually be transferred onto the uninfected red cells, which allow a larger size of rosettes to be formed. So this is what we call the type 3 rosettes. So with that, we decided to move on to test another hypothesized roles of rosetting, which is to form a shield against other harmful extracellular threats besides the phagocytes. And at about the same time, when we were about to test this particular hypothesis, it was during the time when artemisinin resistance was a major issues in the greater Mekong subregions of Southeast Asia. So artemisinin was actually, until now, is still the first line treatment for malaria. However, it is actually losing efficacies because of the massive spread of artemisinin resistance developments by the malaria parasites. So what we like about this artemisinin was that they kill the parasites very fast. So these artemisinins and its derivatives actually work as a prodrug, where upon administrations, they can be activated and they can act as an oxidative bomb against the parasites and the infected erythrocytes, which will actually either directly kill the parasites or they make the infected red cells more rigid, which will facilitate the phagocytic clearance by the host immune system. So when we look at the structure and the mechanisms of killing actions by artemisinins, we immediately link it to our rosetting hampering effect on phagocytosis. So during that time, we wonder whether this rosetting phenomenon may be associated with somehow with this artemisinin resistance development. So luckily for my team, we actually work actively in in one of the research camps in the border area between Thailand and Myanmar, which is SMRU. So this was actually one of the hotspots of artemisinin resistance malaria. And during that time, the only mechanisms associated with artemisinin resistance developments revolved around the mutations of the parasites known as the Couch13 mutations. So what's so special about these Couch13 mutations was that it has already been proven that these mutations actually confer resistance ability to the early stages of the parasites, which makes them resistant to the artemisinin and its derivatives. So the magical bullet artemisinins originally are actually active or effective against almost all blood stages of the parasites, as shown by this schematic diagram here. So the Couch13 mutant parasites works by narrowing the therapeutic window stages for artemisinin, where the ring stages of the parasites become more resistant. So what they do is they allow the ring stages to pause the development when they come across these artemisinins within the circulations. And then these will actually minimize the damage that can be caused by the artemisinin on the parasites, because the ring stages will have a minimal quantity of free ferric ions to actually activate artemisinins, and they will wait for artemisinin to be cleared by from the circulations, because artemisinin has very fast clearance half-life. Then they will resume the maturations or the developments to complete their life cycle. So this is how Couch13 mutants works against artemisinin. But when we look at the picture of artemisinin resistance based on Couch13 mutations, we actually raise a big questions, because if these Couch13 mutations and this ring-mediated strategy is the only strategies used by the Plasmodium parasites to counter the actions of artemisinin, we wouldn't find such a clear-cut difference in terms of a clinical parasite clearance time by artemisinin between the Couch13 mutants and the Couch13 wild-type parasites. Because if you follow this logic of artemisinin resistance picture, if the patients with Couch13 mutant parasites that were presented to the clinic, where predominantly the parasites during that time was actually late stages. So technically speaking, if you give them artemisinin, the parasites within the body of the patients would still be completely killed by the artemisinin. Because if you follow this mechanism, the late stages of the parasites are technically speaking still sensitive to artemisinin. But this was not the case. We managed to see a clear-cut difference in parasite clearance time between the Couch13 mutant and Couch13 wild types parasites, regardless of the predominant stage of parasites harbored by the patients upon the time of admissions. So obviously, the late stages parasites must have some sort of strategies that we have overlooked all this while that can actually counter the actions of artemisinins. So we decided to look at this matter from the perspective of rosetting. So what we did was we first proved that artemisinin could actually facilitate the parasite clearance of the infected erythrocytes via phagocytosis. So what we did was we purified the infected red cells from the culture, and we exposed them to artesunate, one of the derivatives of artemisinin, and then exposed them to the phagocytes. And we found that by doing so, the artesunate exposed parasites, in fact, become more susceptible to phagocytosis. Okay, and when we tested with more clinical isolates to see whether they can actually respond to increased concentrations of artesunate, we found that majority of these clinical isolates from the Thai-Burmese border actually has rosetting machinery that can respond positively to the presence of artesunate. And when we further divide these parasites based on the susceptibility level to artesunate, where we have this artesunate resistant parasite group and the artesunate sensitive group, we found that the parasites that are resistant to artesunate actually show faster and higher degree of artesunate mediated rosette stimulations, okay, as shown by this set of graph here. So obviously, their rosetting machinery respond differently to the presence of drugs. So then, when we no longer purify the infected cells, we expose the parasites mixture with uninfected red cells straight away with the phagocytes, where they are allowed to form rosettes with the uninfected red cells. We found that this time the artesunate resistance groups actually show lowering of phagocytosis susceptibility. But when we purify the infected red cells so that there is no uninfected red cells in the vicinity of the parasites for them to form rosettes with, the resistance parasites now become susceptible to phagocytosis again, following the exposure to artesunate. And we managed to tabulate that, in fact, the more phagocytes are actually needed to phagocytose a rosette forming infected erythrocytes. And then we further proved that the artesunate resistance parasites with the ability to form rosettes are in fact managed to survive better following exposure to artesunate. However, when we remove their ability to form rosettes by removing the ligands needed to form rosettes, the resistance parasites then become susceptible again to exposures of artesunate. So this further proved that the rosetting machinery of the parasites is involved with conferments of artesunate resistance mechanisms. But when we look at the expression profiles of rosetting ligands, which takes hours to complete, it doesn't tally with our discovery that the artesunate resistance parasites actually are protected against the drug by forming more rosettes in a faster manner, because the whole process from transcriptions all the way to expressions on the surface of infected red cells, as shown by this cascade here, takes at least 6 hours to complete. But our rosette stimulations by the resistance parasites can be completed within minutes. So obviously, the stimulations does not happen or is not attributed to the transcription stimulations. So then we studied further and we suspect that it might be at this point where when the parasites are exposed to the drug, the warehouse structures, which is what we call Maurer's craft in the parasites, may actually be triggered to release the rosetting ligands immediately onto the surface of the infected red cells. So then they will not need to synthesize the rosetting ligands from scratch, which will take hours to complete. Now, in order to test this, we decided to actually use one particular approach where this particular trafficking machinery is actually requiring the cholesterol in the whole cell membrane. So without the whole cell membrane cholesterol, the Maurer's craft trafficking machinery will be completely blocked. So we attempted to exploit these particular features of the trafficking machinery by depleting the cholesterol of the red cells membrane, and then subjected them to artesunate exposure to check the rosetting machinery of the sensitive parasites and the resistance parasites. And in fact, when the membrane cholesterol of the infected red cells is depleted by this MBCD, which is a chemical that can actually get rid of the membrane cholesterol, we found that the resistance parasites on the right no longer have this ability to respond positively towards artesunate exposure, where their rosetting machinery become inert. So this actually indirectly show that the trafficking machinery of Maurer's craft is crucial for the resistance parasites to actually increase their rosetting following exposure to artesunate. And then we decided to fine-tune which exact rosetting ligand by the parasites is actually involved in these artesunate-mediated rosette stimulations. And we found that it was actually PFEMP1 that actually get involved in these artesunate-mediated rosette stimulations. And this, in fact, tallied with our discovery that the late ring stage of the parasites actually respond positively to the exposures of drugs by forming more rosettes. And at this stage of the parasites, the only rosetting ligands available on the surface of infected rat cells is actually PFEMP1. So with that, we then decided to look at the genes associated with this phenomenon. So out of the hundreds of genes associated with these rosetting ligand expressions, we shortlisted 2 gene candidates, and then we tested the rosetting assay by recategorizing our clinical isolates, and we found that in fact the mutations, specifically the deletions of of these 2 genes, actually conferred uh, superior rosette stimulations ability to the parasites when they are exposed to the artesunate. And then when we did this bivariate analysis by studying the correlations between these gene mutations with the Couch-13 mutations and the time point when we started to discover the drug resistance issues, we found that in fact these mutations on these 2 genes stated here were strongly correlated to the events of Couch-13 mutations and the discovery of drug resistance issues in the clinical isolates from the Greater Mekong subregions. So with that, we published our discovery where rosettes actually contributes towards protections for the infected rat cell late stages against artemisinin and its derivatives. So it serves as a mechanism, protective mechanisms for the parasites to buy more time to complete their maturations when they come across artemisinin, so that they can actually reinvade into ring stages where they can use a more stable and more specific strategies like the Couch-13 mediated strategies to counter the actions of artemisinin. And not long after our discovery, a separate team from China actually found similar discovery where they actually knocked out this PFEMP1 and found that the knocking out of this gene actually restored the resistance parasites susceptibility to artemisinin. And subsequently, another team from Japan also found that the cytoadherence phenotypes of the parasites is actually correlated to the development of drug resistance. And then we also tested the effects of rosetting stimulations with other mainstream anti-malarials used in the market. And we found that actually the drug-mediated rosette stimulations is not specific to only artemisinin. And we suspected that we actually speculated based on our results that the rosette mediated the drug mediated rosette stimulations may actually serve as a non-specific or very crude strategy by the parasites to counter the actions of drugs, so that they can actually get more time to develop a better mutations to come out with a more specific strategies against a particular anti-malaria drug. So it's a little bit like our innate immune systems, where the strategies are non-specific but very fast, and then it will buy time for our immune systems to develop the more specific adaptive immune responses. And in fact, an earlier studies actually shows evolutionary correlations between the cytoadherence gene genotype selections and the selections of genotypes associated with drug resistance, not only for artemisinin, but also for other drugs such as chloroquine. So how about the zoonotic parasites like Plasmodium knowlesi? So this has actually become the major challenge faced by the healthcare systems in Malaysia. So Malaysia has successfully cleared the local transmissions of human malaria parasites for many years. However, at the moment, we have been plagued seriously by Plasmodium knowlesi, where we have persistent transmissions of knowlesi malaria in human. And um, one not so nice thing about this zoonotic malaria is that knowlesi malaria is actually potentially fatal to humans. And death cases have actually been reported from time to time. So this zoonosis is not something that we can take lightly on. So of course, not everyone has an equal risk of acquiring knowlesi malaria. It actually depends on where you are and what kind of occupations that you are actually doing. So those that are associated with forested areas or living in the forested area or fringe of forests are at particular high risk of acquiring knowlesi malaria because the Anopheles mosquitoes in this area may actually bite on monkeys and humans. So they are showing a broader feeding preference of hosts where they are not specific to only monkeys. Okay, this is what we call the zooanthropophilic preference of biting behaviors of Anopheles. So in that case, they can actually transmit pathogens from monkeys to humans and vice versa. So we also characterize the cytoadherence properties of Plasmodium knowlesi infected erythrocytes, and in fact, we were the first group to actually discover the rosette phenomenon of Plasmodium knowlesi. And we also characterize the endothelial cytoadherence profile of Plasmodium knowlesi. And one interesting thing about Plasmodium knowlesi is that they are actually not sticky to naive endothelial cells. So the endothelial cells have to be primed and exposed to the Plasmodium knowlesi antigens for a while. Then only they will become highly sticky to the infected erythrocytes. And they show specific preference or tropisms of binding to endothelial cells derived from certain organs, such as the lung and the kidney, but not to the brain. And this actually agree with the reported severe complications caused by Plasmodium knowlesi, where we usually see manifestations of severe knowlesi malaria in the form of acute respiratory distress syndrome and acute kidney failure instead of cerebral malaria. And we also characterize the receptors involved in the cytoadherence of Plasmodium knowlesi to endothelial cells derived from different organs, where we found that different receptors are actually involved in the binding events with the Plasmodium knowlesi. And recently, we found another protein from human known as periostin or OSF2 that can also mediate rosette stimulation by Plasmodium falciparum and Plasmodium knowlesi. So their role is a little bit similar to IGF-BP7, where they can also increase the rosetting rate by Plasmodium falciparum and Plasmodium knowlesi. And at the moment, we are actually looking into the role of periostin in disturbing the equilibrium of cytoadherence dynamics of the malaria parasites, because we found that the exposure to this protein actually reduce the parasite's likelihood to adhere to the endothelial cells while increase the rosetting rates. And in that case, it may actually disturb or change the outcomes of malaria, where it may actually reduce the likelihood of the host to acquire severe complications of malaria should the level of such proteins is actually higher within the particular host. But we are doing more research to actually prove this. So the bring home message from this talk is that the cytoadherence phenomenon by Plasmodium infected red cells actually play important roles in malaria pathogenesis. And the dynamics between the different types of cytoadherence events mediated by the infected erythrocytes may actually change or determine the outcomes of malaria. And our understanding on rosetting phenomenon by Plasmodium infected erythrocytes has significantly improved over the past 20 years, where we actually found that rosetting can actually enhance parasite survival by helping them to evade our immune system via either hampering phagocytosis or assisting the sequestrations of the parasites away from the splenic filtrations. And they also protect the parasites against other extracellular traits such as the anti-malarials. And to date, rosetting phenomenon is found in all medically important malaria parasites. And in fact, with simian malaria parasites, we found that rosetting phenomenon is actually very prevalent. In some species, for example, Plasmodium courtneyi and Plasmodium fragile, the rosetting prevalence is actually very high among the parasites from the monkey isolates. So this may be something that can serve as an interesting research topic in veterinary science. And with that, I also like to acknowledge the team members and the funders that have been very supportive through the years for the research that my team has conducted through the years. So with that, I thank you for your attention. And I'm ready to answer whatever questions that you have pertaining to these particular presentations. Thank you.
Thank you, Dr. Lee. It is a very interesting presentation, and I think it's meaningful for clearance malaria. And let me resume your presentation in bahasa because it's my student. Jadi para mahasiswa, eritrosit yang terinfeksi oleh Plasmodium itu mengeluarkan suatu protein. Dan dengan adanya protein ini kemudian membuat lebih lengket sehingga dia bisa membentuk roset lengket baik yang kemudian yang di tengah maupun yang di sel endotel gitu kan, sehingga kemudian dia bisa, dan pembentukan roset ini dia cara parasit untuk menghindari dari fagositosis. Nah, selama ini pengobatan malaria itu dengan artemisinin, tetapi sebagian besar itu sudah mengalami resistensi. Ternyata resistensi ini juga berhubungan dengan pembentukan roset tadi. Jadi artemisinin ini bisa bisa mematikan plasmodiumnya melalui fagositosis. Padahal tadi dengan adanya membentuk roset tadi dia susah untuk difagositosis. Artinya ini adalah nantinya dengan penemuan dari Dr. Lee yang kemudian fenomena roset ini diharapkan nanti pengobatan dari malaria itu diarahkan ke sana untuk menghilangkan roset-roset ini sehingga artemisinin masih bisa. Itu juga kemudian berlaku pada Plasmodium knowlesi, yang dimana Plasmodium knowlesi itu pada primata yang juga zoonosis. Itu yang bisa saya simpulkan dari tadi apa yang disampaikan oleh Dr. Lee. Sekarang saatnya diskusi. Saya mengundang para student untuk ataupun attendance yang lain untuk menyampaikan pertanyaan. Silahkan raise hand atau menuliskan di chat box. Silahkan.
You can also ask in Bahasa Indonesia. I try my best to understand the questions.
Okay. Thank you. Silakan para mahasiswa atau mungkin para dosen dulu kalau ada yang mau bertanya. Prof. Endang, Dr. Mufasirin. Okay. In the chat box, Dr. Mufasirin asked Dr. Lee presentation is, sorry ya.
Okay. So I saw one question
Yes,
on the chat box.
yes, from Dr. Mufasirin.
Okay. Thank you, doctor, for the questions. So many years ago, there were actually several teams from different countries. The nearest one was actually the researcher from Thailand. They actually attempted uh, several anticoagulant as the adjunct therapeutic agent to dissociate rosettes with the hope that it may actually reduce the severe pathogenesis of malaria. However, it was very complicated because a lot of these chemicals that can serve as coagulopathy side effects to the patients. So it's very difficult to actually fine tune the safe dose to be used on all patients. So the even if there is any therapeutic value conferred by anticoagulants like EDTA or heparin and quaternary sulfate were the components being tested by the Thai researchers back in the 90s. So even though these chemicals may actually produce some sort of a protective effect, it is very difficult to fine-tune the right dose that is safe to be used on all patients because the therapeutic window is very narrow. You just need to tune the dose a little bit higher, then the patients may actually die because of coagulopathy associated side effects. So this is still a very tricky issue to address. However, we are actually trying to explore whether we can use protein candidates like IGF-BP7 as an adjunct therapeutic agent to address these particular issues. So works are still ongoing. Um, so far, we have tested on mouse malaria model. The result is encouraging, but it doesn't show 100% protections. Yeah, I hope I answer your questions.
Thank you, Dokter. Yeah.
Silakan mahasiswa. Okay, before student ask the question, maybe I will ask Dr. Lee about preventing phagocytosis. Are rosettes also associated with
90s research group led by Professor Udong Sampet from Mahidol University. I think she retired already. And I think Professor Cassini Chotiwanit, also from Mahidol University, they did a lot of these very deep characterizations and association studies between rosetting and cytokine profiling. If I didn't remember wrongly, but I have to double check, there was no clear association between the rosetting ability of the parasites and the profiling of cytokines from the patient's serum. However, I would like to emphasize that a lot of these characterization studies are more like a snapshot method where we take the sample and then we quantitate the level of cytokines and certain markers from the blood samples. But when we take the samples from the patient, we do not know for how long the pathogenesis has actually happened. So the snapshot may not be able to tell us a lot on the time-based progress of the pathogenesis. So whether the rosetting phenotype, which is associated with pathogenesis of malaria, can actually influence the release or the productions of these markers or cytokines, I think we need to do a more systematic approach of investigations to further validate this. Yeah, I hope I answer your questions.
Thank you. Para mahasiswa? Belum ada? Kalau belum ada,
Lanjut
mungkin
ini.
I have one question more. It's a phenomenon rosette. It's a rosette phenomenon also occur in Plasmodium berghei in mice.
Yes,
Okay.
yes, they are. We actually we did it. We did this experiment with mouse malaria model. When I was in Singapore. So we tried with Berghei, Yolii, and no, I only try on Plasmodium berghei and Yolii. Yes, they do form rosettes. But the prevalence rates are different. Yeah, but rosettes are available. Yeah. I saw Lim
Okay, thank
raising
you.
hands,
Yes.
is it? Yeah.
Yes. Li Jingho, raise hand, please. Lim Jingho.
Yeah, so maybe before other mahasiswa want to ask question, maybe I'll ask a quick question. So because just now Dr. Lee mentioned about Plasmodium knowlesi in macaques, right? So I want to ask, do we see any specific different changes of the stage or the specific host preference between macaques and humans?
Okay, that is a very good question. Um, so we actually did some studies on this, where when we try to transfer the parasites, the Plasmodium knowlesi that were isolated from human, back into the monkey, uh, it is very easy. But then once the parasites entering the monkey blood cells, for you to actually transfer it back into the human red cells, it can be quite difficult. So obviously there is this tropisms or preference of invasions between the human blood cells and the monkey blood cells. And obviously Plasmodium knowlesi may still prefer the monkey blood cells. But I personally believe that they are adapting well. And whether eventually we can actually sustain transmissions of knowlesi malaria from human to human without the need to have monkeys, I think this is a big question mark. And in fact, last year, the researchers from Thailand actually found that when they feed the knowlesi malaria patient blood to Anopheles mosquitoes, eventually the Anopheles mosquitoes successfully produce viable sporozoites of Plasmodium knowlesi in their salivary gland, which means that technically speaking, the circle of transmissions of knowlesi malaria is completed without the need of monkeys. So in that case, eventually Plasmodium knowlesi may be, just like another human malaria parasites, but with monkey as reservoirs. So yeah, there is a preference, but I think the parasites are adapting.
Thank you very much, Dr. Lee.
Thank
Is there
you.
any students that wanting to ask more questions? You can directly open your mic atau type di chat box. Untuk mahasiswa yang ingin bertanya, bisa langsung on mic atau type di chat box ya.
Dua-dua pun boleh.
Ya, silakan mahasiswa, boleh tanya dalam bahasa Indonesia. Iya, Ratna, please Ratna.
Yeah,
Ratna is a magister student.
yeah,
Silakan, Ratna.
thank you, Prof. Lucia. Thank you, Dr. Lee. Your presentation is really nice.
Thank
It's
you.
very different with my job now, so it's really interesting. My name is Ratna. I'm from Vet Public Health student. I have a question, just a little bit curious. As you mentioned before, that Plasmodium infection is increased during the COVID, right?
Yes.
What the factor that make that things happen? I just curious. And the second questions, from your research, it's if I hope I'm not wrong, artemisinin is tattoo resistant, right? To use to cure the Plasmodium infection. And for the alternative medicine, what is now used in Malaysia to cure that infection? Do you
Okay.
get the question? Okay, thank you.
Okay. Thanks, Ratna, for the questions. So I will answer the first questions regarding why COVID contribute to the increased reported cases of malaria. So I think when you have this big scale pandemic that happens out of sudden, involving so much of fatalities, all countries would definitely devote or dedicate their resources in healthcare system to just to handle COVID. And this happened in all countries. And when you only have that much of resources in your healthcare structure, uh, and you use almost everything just to handle COVID, then definitely other infectious diseases or other healthcare problems will have to be sidelined and become less prioritized. So during the COVID period, um, at one country already faced a lot of challenges, let alone the developing countries and the underdeveloped countries. And majority of the countries affected by malaria are actually developing or underdeveloped countries. So they have no choice but to actually focus just to handle COVID. And when that happened, of course, the measures that they originally installed to reduce malaria transmissions were all stopped altogether.
Mm-mm.
And that allows the transmissions of malaria to increase because you stop dispatching bat nets, you stop spraying insecticides, and you stop monitoring the transmissions in your community. So all these actually contributes to the increased transmissions of other infectious diseases, including malaria, during this COVID pandemic period. And now that the COVID pandemic is considered ending, but even though it's still persisting, we have more time and adequate resources to focus back on other issues that we actually have in our healthcare structure. Then we realize that the cases actually bounce back. And this is not restricted to only malaria, but many other infectious diseases as well. So COVID pandemic actually caused our effort to control the transmissions of many diseases to go backward by at least a few years. And it will take us a few more years of effort to bring back the transmission level to the level before the COVID pandemic. So this is one of the bad consequences of having uncontrolled a pandemic that caught us unprepared. And that is why after the COVID pandemic, you see that more countries are actually putting more effort and investment on pandemic preparedness because we try to avoid another round of pandemic from happening. And it will actually cause other healthcare problems to increase by magnitude. So I hope that answers the first question. And the second question is regarding the artemisinin resistance. So unfortunately, artemisinin is still the first line treatment of malaria,
Mm.
even though artemisinin resistance has been reported and it is actually widespread. Started from the greater Mekong subregions of Southeast Asia and now already spread outwards, and even Africa is actually facing it. Um, so what we are doing now is actually using partner drugs for artemisinin. So we use artemisinin-based combination therapy to treat malaria patients. So we will couple artemisinin with another drug, usually drugs with much longer clearance time, so that it will work synergistically to kill the parasite much more effectively, and with the hope that it will reduce the incidence of treatment failure. But one annoying thing about these artemisinin resistance parasites is that we notice that they develop resistance to the partner drugs very fast. So this lead to the need to actually change the partner drugs for SCT or artemisinin combination therapy relatively frequently. Otherwise, your SCT treatment will also fail. So this is the current strategies used by many countries, including Malaysia. And unfortunately, we don't have a new drug yet to replace artemisinin. Nevertheless, there are a few new drug candidates that are still being tested, and hopefully, in the next few years, they will be available in the market. But unfortunately, usually new drugs means more expensive, and not everyone may be able to afford using that drugs. So this will be another issue later on. So I hope I answer your questions.
Yeah, very clear. Thank you very much, Dr. Lee.
Thank you.
Masih ada para mahasiswa? Cukup? Enough?
Sudah petang ini, sudah petang ini.
Gimana? Cukup? Because no student answer any question anymore. My role as
There,
moderator is complete.
there is one
Ada lagi,
question.
Prof.
Oh,
Lucia,
ada.
one
Mana?
question.
Since the discovery of rosetting in malaria, while
Oh.
far-fetched, do you think developing a vaccine that prevents this phenomenon could potentially lead to a cure for the disease? Or is it a lot more complicated due to the parasite's rapid mutation? Okay, so I think this is pertaining to developing a vaccine against rosetting phenomenon, correct? Now, vaccine development for parasites is always challenging. So we we are not talking about vaccines that are against this particular phenomenon. Just to develop a vaccine that can actually kill the parasites is very, very difficult because the parasites is definitely a bigger pathogens compared to viruses and bacteria. Their genome is much more complex, and they have way more genes and way more innovative way of countering our immune system. So just to have a vaccine candidates that can really work for a long enough time against the disease itself is very, very difficult. And hence, if we have a technology or a know-how to develop vaccines against malaria, we would have prioritized a vaccine candidates that can kill the parasites first, instead of just focusing on neutralizing the virulence of the parasites. I'm not saying that it's like completely impossible, but it is highly challenging. And nowadays, we got several vaccine candidates for malaria. And I know that there is one new vaccine candidate being launched recently. So we shall see whether it really works or not. Yeah, so vaccine developments for any parasitic infections is always difficult. Yeah, unfortunately.
Yes, I am agree with you. Okay, and next one question from Dr. Henny Puspitasari. Very interesting presentation, Dr. Lee. I will ask about the protein that play a role formation rosette, such as PFEMP. Can this protein be isolated to be used as a biological marker for the detection of acute state Plasmodium, which can be used as a material for production of diagnostic kit?
Dr. Henny, thank you for your question. At the moment, there is no group using this PFEMP1 as a candidate for diagnosis or predictions of no, yeah, for diagnosis, no. This is because PFEMP1 is a membrane-bound protein. So it is actually attached to the the surface of the infected erythrocytes instead of being released or secreted into the bloodstream. So if you were to use it as a marker, it may be quite tricky because you may not find it in high level in the serum. So it may not be a reliable marker for diagnosis purposes. And another thing is that PFMP1 is actually coded by a variant gene of the Plasmodium. So that means that the parasites have multiple versions of this protein. So the antibody that you generate may work specifically onto one versions of PFMP1, but may not even bind to other versions of PFMP1. So this makes it very difficult to use a variant protein as a diagnostic candidate, and it also makes it very difficult to generate antibody as a potential candidate for adjunct treatment because this is a variant protein. Even though we know that technically speaking, if you block PFMP1 with antibody, it actually affect the growth of the parasites. Yeah. I hope I answer your questions. Thank you.
Thank you. Masih ada? Cukup? Okay, because karena sudah tidak ada, my role as a moderator is complete. I apologize for any shortcoming. Thank you again, Dr. Lee, for sharing your invaluable knowledge. So I will return the time and screen to Master of Ceremony, Wenn Chyau.
Prof. Lucia, I think there is
Yes.
one more question.
One question. Okay. Thanks. Okay.
Okay.
The last question.
Is whether the adhesion mechanisms of rosetting can be utilized to treat breeding if PFMP protein is isolated and engineered. Okay, this is a good question. We might be able to manipulate the pathogenesis outcome with PFMP1 protein theoretically, but to produce it and use it in treatment, it depends on many factors. For example, the stability of the protein. When you administered it, how long can it stay in the circulation, and whether it will cause non-specific effect that can backfire. So all these require more research to be done before we can actually push it or postulate it as a treatment candidate to reduce the complications caused by malaria. But this is definitely an interesting topic that can be investigated further. Thank you so much.
Masih ada sebelum saya tutup? Sebelum saya kembalikan ke MC?
Baik, Prof. Lucia. Okay, so thank you very much, Dr. Lee, for the insightful presentation and discussion. And I also believe that your presentation has been really inspiring our beloved students. So before we move to the closing, let's have a group photo together. So maybe now everyone can turn on your camera with the virtual background. I'll start with slide number 1. Okay, slide number 2. 1, 2, 3. Slide number 3. 1, 2, 3. Slide number 4. 1, 2, 3. Slide number 5. 1, 2, 3. Slide number 6. 1, 2, 3. Slide number 7. 1, 2, 3. And slide number 8. 1, 2, 3. Slide number 9. 1, 2, 3. Slide number 10. 1, 2, 3. Slide number 11. 1, 2, 3. And the last slide. 1, 2, 3. Okay, so now we have done our documentation. We will move to our handover of e-certificate to our guest lecturer, Dr. Wenn Chyau, Ph.D., which will be presented by Dr. Mufa Sirin, Dr. Hewan, Master in Science. So Dr. Mufa.
Okay, yeah. Thank you, Dr. Lee, for sharing your knowledge with us students. Yeah, on behalf of the faculty, yeah, we thank you for your time and the opportunity, yeah, to share your knowledge with us. Yeah, as a token of our gratitude, we present a certificate as a token of respect to Dr. Lee. Please,
Thank you so much.
Dr. Lee.
Thank you for the invitations. It's a pleasure and an honor to be able to share a little bit about malaria with you guys. And I look forward to having collaborations in terms of research, teachings, and
Yes.
whichever form in academic with you guys in future. Thank you so much.
Thank
You are
you.
welcome. Thank
Yeah,
you.
welcome. Untuk Prof. Lucia, monggo Prof.
Next.
Ada sertifikat ya untuk moderator. Terima kasih sudah menjadi moderator ya, penghubung antara narasumber dengan peserta. Terima kasih Prof. Lucia. Bisa diterima
Terima
nggih?
kasih. Yes, terima kasih.
Terima
And
kasih.
thank you,
Terima
Dr.
kasih.
Terima
Lee.
kasih.
Thank you.
Makasih semuanya.
Ya, oke.
Okay, thank you, Dr. Mufa, and thank you once again, Dr. Lee. Also, students, we have reached the end of our guest lecture on behalf of the Faculty of Veterinary Medicine, Universitas Airlangga. So I would like to express our deepest gratitude to Dr. Wenn Chyau, Ph.D., for sharing his valuable knowledge with us today. Also, thank you to our moderator, Prof. Dr. Lucia Suwanti, DVM, Master in Agriculture, and to all participants for your attention. So I hope this session has been insightful and inspiring for all of us. And I also sincerely apologize for any mistake that I've made today. Once again, thank you and have a good evening. See you at
Thank
our
you so
next
much.
academic event.
Thank you,
Makasih.
Dr.
Thank
Lee.
you.
Thank you.
Ya.
You, Dr.
Thank
Good
Lee.
night, good night.
Sleep for you.
Thank you.
Ya, makasih.
Prof. Puji. Prof. Puji.
Thank you. Serak Serak aku nggak bisa ngomong
Prof. Puji is good
That's
friend
all.
Dr. Lee. Oh, Puji.
Thank you.
Thank you for the invitation.
Thank you, thank you.
Permit me leave from Chung. Once again, thank you
Yeah.
Thank
for
you. I
anyone.
hope the
Yes.
internet connectivity didn't affect
Okay.
the lecture.
Yes.
Yeah, because our
Yes.
internet over here was a little bit bad over the past few days. So yeah, hopefully the presentation was clear.
Nice.
Yes.
Thank you, thank you.
Thank you.
Thank you.
Thank
Good
you,
night,
thank you.
everyone.
Okay.
Night, yeah.
Yes. Night.
Universitas Airlangga, kau selalu di dada. Hadirkan lulusan profesional berwawasan luas. Ayo, ayo,
Terima kasih, yo. Terima kasih.
makin hebat.
Terima kasih dok.
Ya,
Terima
ya,
kasih Prof.
ya.
Kasih ya.
Terima kasih Pak Puji.
Iya, ini serai.
Semoga lekas sembuh sebelum ke Jepang ya. Pokoknya harus sembuh dulu itu ya.
Inovasi kesehatan hewan peternakan sejahterakan masyarakat. Universitas Airlangga, kau selalu di depan. Junjung